Hello to our friends joining us via recording. We are covering our last lab packet, and that is about the special senses. To help us review for the lab final exam, we are tying together our new information about the special senses with some of our old information, which was the cranial nerves and the functions of the parts of the brain. So the first thing we're going to review are some of the cranial nerves that are important for the structures that we're going to be identifying today. One of the special senses we're going to spend a lot of time on today is the process of hearing, or the way we collect auditory information. For my friends here in class today, help me out in the chat. What is the number and the name of my cranial nerve that helps me with the process of hearing, or that collects auditory information? It's one with a not fun name, right? Yeah, so the number on this nerve is cranial nerve number 8. I'm going to give it to you here both in Roman numerals and in normal person numbers, right? Arabic numerals. Cranial nerve number 8. The name of that nerve is called the vestibulocochlear nerve. The vestibulocochlear nerve. Now, this nerve not only helps out with the process of hearing, what else does cranial nerve number 8 help us to do? It does 2 things. Yeah, the other thing that cranial nerve number 8 does is balance, which our fancy anatomy word for that is equilibrium. So cranial nerve number 8, here's my wink, wink, nudge, nudge for you. We talked about it not only helping us with hearing, but also helping us with balance. Let's make sure we know about cranial nerve number 8. It does 2 important things for us. So let me add that to the bottom here. Cranial nerve 8, still called the vestibulocochlear nerve. Here's the thing with this name, vestibulocochlear. If you chop this big long name in half, it helps me to remember the 2 things that this nerve does. The cochlear part of its name refers to the cochlea, which is the part in the inner ear that helps you with hearing. The vestibulo part of its name comes from some inner ear structures that together we call the vestibule, and the vestibule is the part of your ear that helps you with balance. So the vestibulocochlear nerve, cranial nerve number 8, that helps us with balance and with hearing, vestibulocochlear. Let's move on to the cranial nerve that helps us with vision, or the process of seeing. Which cranial nerve helps us with the process of seeing? Name and number. Yep, cranial nerve number 2. Cranial nerve number 2 is my optic nerve, the optic nerve. Remember, and we'll see this on our picture coming up, the optic nerve has that thing called the optic chiasm, which is an X that helps me to get vision information from my right eyeball to the left side of my brain, and stuff from my left eyeball to the right side of my brain. So identifying cranial nerve number 2, the optic nerve, it's going to look like an X. It's going to have that X structure. The other cranial nerves that we needed to provide here are the ones that help with eyeball movement. And I actually told you when we were learning cranial nerves to learn these in a group. Let's start with the numbers. Can we rattle off the numbers really fast of the cranial nerves that help with eyeball movement? Yeah, perfect. We're remembering that list that I told you, right? So the numbers of the cranial nerves that help us with movements: number 3, number 4, and number 6. All right, help me out in the chat. Cranial nerve number 3, what's the name of that nerve? Cranial nerve number 3. Yep, number 3 is the oculomotor nerve. Number 4 is called the Yep, number 4 is the trochlear nerve. That's the smallest cranial nerve. And number 6, my last one that does movement, its name actually sounds like a movement word because that's the movement that it helps the eyeball do. Cranial nerve number 6 is the abducens nerve. So it helps me to move my eyeball away from the midline of the body. Abducens, cranial nerve number 6. Okay, so like I mentioned before, wink, wink, nudge, nudge. Make sure that we know vestibulocochlear nerve, make sure that we know the optic nerve, make sure that we know these movement nerves for the eyeball. If we cover them in more than one packet in lab, there's a very, very good chance they're going to show up on the exam. So there's my wink, wink, nudge, nudge for you. We named our nerves. Now we need to find our nerves. So I'm going to make a quick list for us here. We said that the process of hearing was cranial nerve number 8. We said that vision was number 2. Our eyeball movements, those were 3, 4, and 6. And balance was also number 8. When we are labeling our cranial nerves, remember that we start at the front and we work our way back. And when we get to a group of cranial nerves, we always go from the middle toward the outside. So let's go through here and we'll just label our numbers from front to back. The first thing that I see up here is cranial nerve number 1. We didn't talk about cranial nerve number 1 in our list of things that I asked you about, but what does cranial nerve number 1 help us to do? What does cranial nerve number 1 do? Yeah, cranial nerve number 1 is called the olfactory nerve. That's the nerve that helps us with the sense of smell. Cranial nerve number 1, the sense of smell. Back behind it, we can kind of see that crisscross pattern. That's cranial nerve number 2. Cranial nerve number 2 was one of them that we were recognizing. Number 2 is that optic nerve. So cranial nerve number 2, we can see it here. We would also see it extending. If we had all of it here and they hadn't chopped it off, it would look like this, cranial nerve number 2. So cranial nerve number 2, the optic nerve. Back behind it, in the middle of the pons, is cranial nerve number 3. Cranial nerve number 3 was one of my first cranial nerves that helped me with the process of moving the eyeball. This is the one that's called the oculomotor nerve. So that's in the middle of the pons. Here's the pons right here. Next to it is the smallest cranial nerve. The smallest cranial nerve is cranial nerve number 4. Cranial nerve number 4. This is the trochlear nerve, another movement nerve, and this one wraps around from the back side of the brainstem. We just see it peeking out right here. Hey, this big one right here is cranial nerve number 5. What was the name of cranial nerve number 5? That was the biggest cranial nerve. Yeah, number 5 is the cranial nerve called the trigeminal nerve. If you remember when we were talking about cranial nerves, that was the one where I told you to take your 3 fingers and spread them out wide and put them kind of at the angle of your chin to give you an idea of where it sends information or collects information from. So it's all the way up from the forehead and along the top jaw and along the bottom jaw as well. So cranial nerve number 5, the really big trigeminal nerve. Then we go down into this big mess down here. Since we're moving down, we also move back to the middle. So this one here in the middle is cranial nerve number 6. 6 is my last movement of the eyeball nerve. That's the abducens. Next door to number 6 is number 7. Number 7 is the facial nerve. Facial nerve, it's going to your face. Next to it is number 8. Cranial nerve number 8 is one of them that we talked about that helped us with balance and helped us with hearing, the vestibulocochlear nerve. So we see cranial nerve number 8 here. Then we see cranial nerves number 9 and number 10 right next to each other. Let's see if I can get my numbers distinct enough. Number 9 and 10 are right next to each other. Remember that number 10 is called the vagus nerve, and it goes everywhere. We go down to the bottom for cranial nerve number 11, the accessory nerve, and then remember that 12 bounces back up to the middle. So here's our cranial nerves numbered. I know I saw the question in the chat about which picture we'd be using on the exam. I don't know that we're going to use a color-coded picture like this one, but what we might use is a picture that shows the cranial nerves. If you do a Google search for cranial nerves unlabeled, I believe there's a picture that shows all the cranial nerves in yellow as opposed to all having different colors. So you can try referencing that. Obviously, I can't give you a model in front of you and ask you which cranial nerve we're looking at. So like I said, do a Google Images search for cranial nerves unlabeled, and you can find some good pictures to practice on, like this one that had them color-coded, but also ones that have them all there, just not color-coded to be different. So the ones that we're focusing on today that are important to our special senses we're talking about: the cranial nerve for vision, number 2, cranial nerve number 3, which helps us with movement, number 4, that's doing movement, and number 6, that's doing movement. And then cranial nerve number 8, right here, helps us out with hearing and balance. Those are our cranial nerves of the day. Last thing to remind us of are some of the places in the brain where we process this kind of information. When we talk about 2 things that are processed in a particular lobe, which lobe of the brain does auditory information? Which lobe do you hear in? Yeah, we're hearing in that temporal lobe, the temporal lobe of the brain. When we talk about the process of vision, which lobe of the brain do we see in? Have my friends all fallen asleep? Jesse's awake with me. Everybody else has gone quiet. We're right. Yeah, Jacqueline said basically I've fallen asleep. Yep. The lobe of the brain that I process visual information in is the occipital lobe, there in the back. Then we talk about the parts of the brain that do particular things. So I know we had some questions about this one here, but here's my first hint. When we talk about movement, which lobe of the brain, let's pretend it wasn't a part question, let's pretend it was a lobe question. What's the only lobe of the brain that helps me with movement? Which lobe helps with movement? Yeah, the only lobe that does anything motor is the frontal lobe. When I ask you for the particular part of the frontal lobe that's doing movement, there's 2 answers that we could give. The part that comes from the lab packet in particular is what's called the precentral gyrus. The precentral gyrus, the bump in front of the central sulcus. When we talk about the more lecture-specific structure, the way that we talked about what's inside the precentral gyrus, we're absolutely right in the chat. We call that the motor homunculus. So either of these structures would be correct. When you're looking at it on the lab exam, that's just going to talk about the precentral gyrus. It's going to be a little bit more general. But like you saw on the lecture exam that we took, the motor homunculus is the specific place where we talk about eyeball movement. Remember, by the way, when you were looking at that map and it showed the eyes in there, we had a lot of motor information that went to the eyeballs. That's what allows you to move your eyes so specifically. The eyes were really big on that map. So not only can we blink, but we can move them up and down, side to side. We can do all kinds of different things with our eyeballs. That's because they're so large on the motor homunculus. Yeah, roll your eyes, Jesse, right? We're doing some eye rolling right now. Let's be real. It's that part of the semester. All right, last thing. When we talk about the part of the brain that's helping you with balance, this is also the part of the brain from lecture that we said was easily influenced by alcohol. What's the part of the brain that helps you stay balanced? Yeah, exactly. The cerebellum. So a couple of things that take place in particular parts of the brain, and then we've got our lobes of the brain that we see here that are helping us with some of our special senses or regular movements of the day. So a little bit of review to get us ready for that upcoming lab exam. Let's dive into the new material from this week's lab packet. The first part of this lab packet is about the processes of hearing and the structures of hearing. When we're working on identifying the structures of the ear, which are the structures that help with hearing, there are three regions of the ear. We have what's called the external ear. Let's try again. External ear, we have what's called the middle ear, and then we have the internal ear. There's a part on your lab packet that asks you to sort the structures into their regions of the ear that they're found in. I would encourage you not to take too much time going through and building that list. That's more of an application activity for you. So we want you to make sure that you remember where each of these structures are found, which of these regions they're found in. If you know that information, you don't have to build yourself an exhaustive list. So that's one of those things. We're to the point in the semester, right, where if there are any corners we can cut, we want to try to cut them. We want to save as much time as we can. So don't build yourself a super exhaustive list of where things are found. Just make sure we have an idea of where they're found. That should be enough for us. When we talk about the external, middle, and internal ears, one of the things that will be really important for us to know is what divides up each of these ear regions. So let's start by talking about what divides the external ear from the middle ear. Draw a little line up here, dividing those structures up. The structure that divides the external ear from the inner ear, or from the middle ear, excuse me, is called the tympanic membrane. The tympanic membrane. We can see the tympanic membrane right here on our model. So the tympanic membrane is the dividing line between the external ear, which is everything that you can see, everything that you could touch, from the middle ear, which is what's on the opposite side of the tympanic membrane. So the tympanic membrane is directly attached to some small things called the ossicles. The ossicles are the smallest bones in the body. We can kind of see them right here. We'll see them more clearly on our other ear model picture. So I'll label a couple of them here and we'll see them better in a moment. We have one ossicle, one ear bone that is always found right on top of the tympanic membrane. The name of the ossicle that's right on top of it, that's literally attached to it, is called the malleus. So the malleus is the bone right here that is attached to the tympanic membrane. If you've ever heard of the instrument called a timpani, that's a type of drum. So the malleus is like the mallet that you would use to play that drum. The malleus is attached to the tympanic membrane. Right next to it, the next middle ear bone, or the next ossicle, is a bone called the incus. So the incus is shaped kind of like an anvil, which is that thing for any old time cartoon friends, that thing that Wile E. Coyote and Roadrunner were always perpetually dropping to land on Wile E. Coyote, a anvil. So it's shaped here in the middle, the incus bone. The last one that's hard to see on this picture is one called the stapes. We'll see it really well on my other picture, but I'll draw for you. The stapes looks kind of like this, the stapes bone. So when we peek at our picture, we can kind of see part of it right there at the end of the line. I won't label the stapes on this particular model because this picture doesn't show it well. So we have the malleus, the incus, and the stapes. These are structures that are all found in the middle ear. The other structure that I'll mention in the middle ear for you is this area right here. This area right here is called the pharyngotympanic tube. Pharyngo means pharynx, which is your throat. The pharyngotympanic tube is the tube that goes down into your throat. It connects your throat to your middle ear. The good thing about the pharyngotympanic tube is this is how we equalize pressure between the middle ear and the external ear. So when you go up in an airplane and you feel pressure in your ears and you swallow or you chew gum, the goal of that is to help move the oxygen that's here in the middle ear to equalize the pressure on the two sides of the membrane. That's the good thing about the pharyngotympanic tube, that it would allow you to equalize pressure. The bad thing about it is this is also the way that if you have a sinus infection, this is the way that it can turn into an ear infection. So this tube right here goes into your throat. If you have drainage into your throat, bacteria can actually climb up this tube into your middle ear and give you an ear infection. So this is the tube right here that gives us ear infections because stuff gets from our throat up into our ear. Normally the middle ear part of your ear is filled with air, unless you have an ear infection, and then it's filled with pus or bacteria, and that's not so fun. So other structure of the middle ear, this pharyngotympanic tube. The internal ear is some of the stuff that we see here in the back. The structure that divides up the middle ear from the internal ear is something that we call the oval window. The oval window. And I don't think we actually make you label that on any of our models except the internal ear. So don't worry about labeling this one. It's hard to see. It's a membrane that the stapes, that last little bone, is sitting on top of. On the other side of the oval window, we have fluid for sound waves to be sent into. So on the other side of the oval window, we go into the structure right here called the cochlea. The cochlea. The cochlea, like vestibulocochlear nerve, this cochlea back here kind of looks like a snail shell, and this is where we collect sound information. So you don't actually hear sound waves until they get to the cochlea. This is where you'll perceive them. When we talk about the vestibulocochlear nerve, we said that it did hearing here in the cochlea. The rest of what it does is balance, and balance is done by a group of structures. I'm going to put a little bracket around them. A group of structures called the vestibule. So all of the stuff right here that I've circled is the vestibule. Let's get rid of that label. Oh, not that one, sorry. Get rid of that and draw my circle again. All of this is the vestibule. The vestibule has two parts. Let's see if we make you label them. We make you label part of them. So see these little loops that are up here? These three loops that we see sticking up are things called the semicircular canals. Semicircular canals. Their job is to help you with balance, with equilibrium. So the semicircular canals are these three loops. We'll see them better on a model just of the internal ear. The three semicircular canals help you to keep your balance. The only things that we have not labeled are structures of the external ear. So structures of the external ear, We have a tube right here. Can anyone help me out in the chat? Which of those things we have left has a word in its name that means tube? Yeah, the word that means tube is meatus, right? That was a tube. So here is our external acoustic meatus. The external acoustic meatus. This is the place where you're not supposed to put Q-tips, right? So the external acoustic meatus is the ear canal, basically. When you're doing the process of hearing, ear waves get funneled into this external acoustic meatus, and they transfer to the tympanic membrane where we do bouncing to hear those sound waves. The way that sounds get into the external acoustic meatus is because of this outer part of your ear called the auricle. If you remember way back from week number 2, we talked about the auricular region. That was the ear region. That's where that term comes from. The auricle is the part of your ear that you see. Down below it, hanging down, is the lobule. So the two places that you can get your ear pierced, the lobule down low, the auricle up high. So the auricle, the lobule, and the external acoustic meatus, those are our primary external ear structures. Once we hit the tympanic membrane, now we've transitioned into the middle ear with the ossicles, which was the malleus, the incus, and the stapes. The stapes is found next to that thing called the oval window, which helps us to send sound waves into the cochlea. Here's my other ear model that shows a little bit more clearly what's going on in the internal ear. So Nicole, to your question, I would say don't say that the tympanic membrane is part of the middle ear. It's the dividing line between them. What's the format of the question that asks about whether it's part of the middle ear? Is it a select all, or is it a sorting question? What kind of question was it? So asking for all the parts of the middle ear. So maybe go ahead and select it and see. We might say that it's part of the middle ear and the external ear, or if you stick around after class today, I can check and confirm on that. The tympanic membrane is the dividing line between the two parts, so I'll check to see if it's supposed to be in both parts, maybe. Okay, Jacqueline's saying it is the middle. Yeah, so go with it being part of the middle because the malleus is attached to it. That's probably a good way to think about it. All the same structures visible on this model, but I'll point out that little bone we couldn't see before. There's that bone called the stapes. There's the stapes bone. We can see the malleus and the incus. So the malleus, the one that's attached to the membrane, the incus, the one that's in the middle, and the stapes, that little one. Stapes means stirrup, like a stirrup where you put your feet when you're riding a horse. Malleus, incus, stapes. Then we go into, here's that snail-shaped cochlea back here, and we have those semicircular canals that you can see up here. There's 3 of them, and that I can see here. We have a model that shows this internal ear part up close and personal. That's what you're looking at right here. So when we look at the inner ear or the internal ear more specifically, now we can see a few extra structures that we couldn't see before. We'll start with the ones that we have seen. The first one that we have seen is our little snail shell part right here. Here's the cochlea that I see here. Next to the cochlea, another structure that we have seen are these semicircular canals, these loops. There are 3 of them because these semicircular canals are the things that help you with what's called rotational equilibrium. rotational equilibrium. So your head can rotate in all kinds of different directions. By having 3 different semicircular canals, we know exactly which way our head is rotating in. So these 3 semicircular canals, each of the semicircular canals have a place where it's a little bit wider. So see that wider space right here? We've got a wider space right here. If I look at the opposite side of the model, here's one of those wider spaces and another wider space. These wider spaced areas, I'll circle them for us here. These are the things called the ampulla. We're going to talk more about the ampulla in lecture, but this is the specific place inside a semicircular canal where they actually detect rotational equilibrium. So there's fluid everywhere inside these semicircular canals, but the special neurons that detect the movement of that fluid or that detect your rotation is found here inside the ampulla. So the region where we detect that rotation. Jacqueline says that the word ampulla reminds her of Oompa Loompa. Yeah, a swollen human, right? Yeah, so a little swollen area like those Oompa Loompas. So the other thing that we'll look at here next to those Oompa Loompas, right, those ampulla, are these 2 fluid-filled sacs that I see right here. This one down here in the bottom is something called the saccule. And what we find up above it is a thing called the utricle. We're going to talk again more in lecture about what the saccule and the utricle actually do. But these are the things that do what's called static equilibrium. That is helping you to know if your head's going forward or backwards. Basically, I like to joke, and you'll see this in the lecture lesson, static equilibrium is the way that you're most likely to fall asleep in class. So when your head's going forward and you're falling asleep, static equilibrium is supposed to help you catch that and not fall asleep. Or if you're like completely passed out and your head goes backwards, right, and you're snoring in class, static equilibrium is supposed to stop that too. So forward and backward movement, that's the saccule and the utricle. When your head is rotating different directions, that's rotational equilibrium. Rotational equilibrium has to do with these semicircular canals that have their neurons inside those ampulla, those big, large areas. Let me get my circle here. All of this mess that I see right here, all of this is the vestibulocochlear nerve. What I like about looking at that nerve on this model is you can see that it's made of 2 different parts. This part over here comes from the ampulla and it comes from the saccule and the utricle. This is the vestibulo part of that nerve. And over here is the part that comes from the cochlea, the snail shell. So the vestibulocochlear nerve is where both of these things come together and travel out of the ear to go back to the brain to be processed. So the cochlear part of the vestibulocochlear nerve, the vestibular part of the vestibulocochlear nerve. And remember, all of the things that we see here, all of this is internal ear stuff, the anatomy of the internal ear. How do we feel so far? Thumbs up, thumbs down. Yeah, go for it, Jesse.
So in the packet, when it shows these views, it has us label the oval window. So where would that be located?
Oh, perfect. I had forgotten that structure. Yeah. So the oval window is, if you see this little, let me get black back so you can see it better. You see this little thing down here, this little pink membrane? That is the oval window, So this is the place, if we had our middle ear still around, we would see the stapes, that stirrup-shaped bone, it would be resting right on top of here. So yeah, the oval window is that last structure. It's this little thing right here. So sound waves will come into the stapes that'll bounce basically against this oval window, and it'll send those vibrations all the way through the cochlea here for us to be able to hear them into the snail shell place. So yeah, thank you for reminding me. That one didn't make my list. So there it is right there. Here's a picture that remind me, we do have this in the guided lab lesson, right? Did we have this picture? I'm hoping. Yes, okay, perfect. Because I know what I was going to say, if it's not in the lab lesson, I know it's in the lecture lesson. So this is a really great image to study from because it shows you the anatomy, but also the physiology of the process of hearing. So hearing starts in the external ear with sound waves in the environment. Those sound waves come and bump into the tympanic membrane, which is right here. Remember, the tympanic membrane is kind of like a drum. So we have a drum that's bouncing back and forth because of the sound waves in the environment. As this part of the drum bounces back and forth, that makes the malleus, which is attached to it, start wiggling back and forth, which makes the incus, which is attached to that, wiggle back and forth, which makes the stapes, which is attached to that, wiggle back and forth. The stapes is right next to the oval window. When the stapes is bouncing back and forth, then it causes the fluid that's inside the cochlea to start moving as well. So the sound waves go into the fluid. If we look at the cochlea, there's actually 3 different fluid-filled places inside of it. And we do want to know these 3 fluid-filled spaces. They are different and they each do different things. So the first fluid-filled space that sound waves go into is something called the scala vestibuli. The scala vestibuli is this first fluid-filled tube on the top of the cochlea. Sound waves come into the scala vestibuli and they vibrate all the way down through it until they reach the place in the ear, the physical location in the ear where you would be able to hear them. When they get to the right place where you'd be able to hear them, those sound waves end up vibrating into this middle tube. This middle tube is called the cochlear duct. So the cochlear duct is the tube in the middle of the cochlea. This is where your neurons live that do the process of hearing. So sound waves start out here in the scala vestibuli. They're bouncing around, bouncing around, bouncing around. They find the place where they would be able to be heard. So they vibrate into the cochlear duct. They're heard in the cochlear duct, but we only want to hear them once. We don't want to hear them repeatedly. So once the cochlear duct has heard the sound waves, it releases those sound waves into a thing called the scala tympani. And I like to think about the scala tympani as the place where sound waves go to die, or maybe you can think of it as recycling or the garbage disposal, whatever we want to call it. Sound waves leave the cochlear duct so that we don't keep hearing them. They're going to bounce and bounce and bounce around in the scala tympani all the way until they get to a membrane called the round window. The round window is just like the oval window, except there's no bones on the outside of it. So when sound waves get to the round window and they start bouncing out into this space, there's nothing there to catch them. There's nothing there to hear them. So scala tympani is where I go to get rid of sound waves after I've heard them once, so I don't hear them forever. I put them in the scala tympani. That spits them back out into the middle ear, and I won't hear them anymore. So the process of hearing goes from the external ear through the ossicles of the middle ear into the 3 fluid-filled tubes of the internal ear. First, scala vestibuli, then into the cochlear duct in the middle, then into scala tympani to get out of the cochlea. I see a note in the chat from Hannah that she thinks this particular picture might only be in the lecture lesson. So check that out. I know it's in the lecture handout, and I know it's in the guided lesson as well. So if it's missing from the lab guided lesson, check out lecture, because I know it's in there for sure. To help us with the model we're getting ready to look at, I want you to imagine with me that my little pointer here is now a knife, and I'm making a cut right here. So I'm slicing all the way through. And this slice that I made, I'm now going to turn it around 90 degrees so that we can look at this tube in a different perspective. So let's imagine that we sliced the cochlea, we turned it in a circle, and now we're seeing this. This is the 3 tubes of the cochlea, but instead of seeing them from the side view, now I'm seeing them from the front view. When I look at them from the side view, that's going to help me to label the particular parts of the cochlea. We have 3 fluid-filled tubes when we look at the cochlea. Let's do some numbering so I can make you type numbers and not big long names. Number 1, number 2, number 3. Let's start with the cochlear duct. That was my first fluid-filled tube. Which number on my picture shows me the cochlear duct? Which of these is the cochlear duct? Okay, perfect. So cochlear duct is number 2. Let's see if I can fit its name inside there. Yeah, so Jesse has a note for you, Hannah, that picture was actually in the guided lesson, just not in the lab packet. Cochlear duct is the middle fluid-filled tube. So is it number 1 or number 3 that we would call scala vestibuli? Which is the one that sound waves go into first? Any guesses? 1 or 3? Yeah, so sound waves first go into the thing called scala vestibuli, and scala vestibuli is the top fluid-filled tube inside the cochlea. Sound waves bounce around here in scala vestibuli, then they'll bounce into the cochlear duct when they get to the place where they can actually be heard. Once we've heard them, we send them down here into the bottom area called scala tympani. Scala tympani. There are membranes that divide up scala vestibuli from the cochlear duct and the cochlear duct from scala tympani. Let's talk about those membranes. Well, actually, we don't have to know both of them. We just need to know this membrane right here to divide them up. This one right here is found in the bottom or the basement of the cochlear duct. This one right here is called the basilar membrane. The basilar membrane. Remove those words. There we go. The basilar membrane divides up the cochlear duct from the scala tympani, and it's also the place where these cells that do the process of hearing are standing on top of. Now, these cells that I see standing on top of the basilar membrane are called hair cells. They're called hair cells. It's a special kind of neuron, hair cells. They get their name because on top of them are little hairs. So these little white lines that you see right here are the hairs that hair cells use to hear sound waves. Let me draw some lines here. Here's my little hairs. Those little white things that are inside of there, they're on top of hair cells. The way that hairs can help me to hear sounds is because of this thing I just circled. This thing I just circled is something called the tectorial membrane. The tectorial membrane. Think of the tectorial membrane kind of like a tent that is above the hair cells. So the way the process of hearing works is sound waves come bouncing, bouncing, bouncing in the scala vestibuli. They bounce into the cochlear duct when it's the right place for them to be heard. Those sound waves bouncing around here in the cochlear duct cause the basilar membrane to bounce up and down as well. So the hair cells on top of it are kind of like monkeys jumping on the bed. They're going up and down, up and down, up and down, except this tectorial membrane does not move. So as those hair cells are bouncing up and down, they're bumping into the tectorial membrane. When they bump into this membrane, that changes their shape. And when their shape changes, they detect that. So you hearing sound waves is because we actually change the shape of the hair cells, or the hairs on top of the hair cells. When I change their shape, I open up ion channels. We're going to talk about this in lecture. I open up ion channels that make these hair cells get excited, and they send that message back to the ear, or to the brain, to be processed. So the tectorial membrane doesn't move. Hair cells bump into it with their little hairs. The basilar membrane bounces all over the place. When we're talking about the basilar membrane, the tectorial membrane, and the hair cells, basically all the stuff that's directly involved in the process of hearing, I call all of that stuff the spiral organ. So I'm going to do a kind of a circle around here. All of this stuff together is what I would call the spiral organ. The spiral organ. You really can't read that. Sorry. The stuff circled in blue. So it includes the hair cells, it includes the tectorial membrane, it includes the basilar membrane. All that stuff that sends sensory information back to the brain to assist you with the process of hearing. I think that's all the labels. Is that correct? Is that all the labels? Okay, perfect. So we have another model that shows, yeah, so Nicole, it would include the hairs as well, because the hairs are attached to the hair cells. So yes, hairs included too. This is one cross-section model of the cochlea. The other cross-section model of the cochlea that we have is a little bit less complete. So you see this picture here. I want to help us with drawing some stuff on it to help it make more sense. To help us understand this picture a little bit better, imagine that there's actually a circle up here. One of my tubes is up there. And imagine that there's a circle down here as well. So this model is primarily focusing on the middle fluid-filled tube. What was the name of that middle fluid-filled tube again? What's the one in the very middle? Yeah, that one in the middle is called the cochlear duct. We can see the cochlear duct most clearly on this picture here. That's the fluid-filled space. But the space up above it would be scala vestibuli, and the space down below it is scala tympani. So they just didn't show these two other fluid-filled tubes, the two other scala. They really just showed the cochlear duct clearly. When I'm looking inside the cochlear duct, I see this thing on top that doesn't move. What was the name of the thing that did not move when sound waves are in there? Yeah, the thing that doesn't move, that my cells bump into, that's called the tectorial membrane. The tectorial membrane. And when I look at my little cells that are jumping on the bed, what's the special name of this kind of cells right here? Yeah, these are the hair cells. There's their little tiny hairs. See their little tiny hairs there? So the hairs on top of the hair cells, all of those hair cells we can see really clearly. See this blue membrane right here? This blue membrane is that basilar membrane, the one that bumps up and down when the sound waves come in. So here is the basilar membrane. Oop, gonna have to move that. Basilar membrane there on the bottom. So all of my stuff, I'm gonna circle it for you here, right here. All of this is the stuff that collectively is known as the spiral organ. The tectorial membrane, the hair cells, those hairs, that basilar membrane they sit on top of. I know it's a little bit weird, but I'll mention for you, when you look at this part of the model that's down in the bottom, it's kind of showing you a cross section of the cochlea. If you take a peek at the snail shell over here, you can see that these 3 tubes are found everywhere inside the cochlea, because here's those 3 tubes again up here in this other part that's curling up. So when we look at the cochlea, when we look at the tubes, it's basically just one big long tube that we spiraled up together, maybe kind of like a cinnamon roll or something like that. They just roll up on each other, and we collect the information with this spiral organ that's always found in that middle tube in the middle of the cochlea. Yeah, Nicole says now she's thinking about cinnamon rolls. That's my fault, right? I mentioned food. It's as bad as donuts, cinnamon rolls. Well, I know I saw in the chat, we were talking though about how, you know, whatever it takes to remember, right? So if that helps us remember that it's kind of like a cinnamon roll, we'll take it. It probably won't help, but it does sound very good, cinnamon rolls. Okay, I think, let me check my slides here. That is all the labeling that we need to do of structures of the ear. Are there particular questions that we still have about the process of hearing before we move on to vision? Let me see something here. Check this out. We got an ear going on. Yes, yeah, Jesse, here's the baby. Yep. He is here and he's awake. Okay, glad to hear that, Christina, that you're feeling better about it. And I mentioned it earlier, and I'll say it again too. Lecture lesson number 13 ties into this lab lesson really well. So always feel free to reference that too. Not only will that get you ahead for lecture, but it will also help you out with the lab stuff too. Yep, so lecture lesson number 13 is very long. Yes, because it's a lot of processes. So I would highly recommend starting it earlier rather than later, for sure. So you can start it while you're working on lab stuff, because that'll get you a little bit ahead. Yeah, I like those eyes staring at me, a little bit creepy, but I like them. All right, so speaking of eyes, let's talk eyes. Fun fact about the eyes, the part of your eye that you can see, which is a lot less than what we see on this model, but the part of your eye that you can see is only about 1/6 of the eyeball. A lot of your eyeball is hidden behind your eyelids, behind your eyelashes. There's a lot of adipose tissue that's inside the eye socket. So we see overall very little of our eye. But the parts that we can see when we are looking at the eyeball or when we're dissecting an eyeball or clearing away some of that extra bone tissue and stuff help us to know how the eye functions in the process of vision. So let's start with a review of some of the bony structures that we have to know for the upcoming exam. So let's do some underlining here. Notice that there are several bones that we're supposed to label on this model. Now, on the exam, to be frank, I'm probably not going to ask you to label these bones on this particular model, but we need to know where those bones are. So we're going to use this model, even though it can be a little more challenging to interpret. If you can find those bones on this model, you are prepared to find them on an easier model. Let's start here with this one. This one over here makes up your nose. This one should be easy for us. What's the bone I see right here? Yeah, that one there in the middle, that is the nasal. bone, the nasal bone. Now, the reason I point out and the reason we started with this one right here is because the nasal bone is medial on your face. It's going to matter that you know medial versus lateral, because when we start naming muscles, notice that we have a lateral rectus muscle and a medial rectus muscle. These muscles look exactly the same, but one of them is on the lateral side and one of them is on the medial side. The only way you're going to be able to tell the difference between the lateral and the medial rectus muscle on this model is by knowing where the medial side is. So find that nasal bone to find the medial side of the face. This is the bone up here that makes up your forehead. What's the bone up here that makes up the forehead? What's the name of the forehead bone? Yeah, that bone up there is called the frontal bone, the frontal bone. Underneath your eyeball, it makes up the top part of your jaw. What's the name of the bone that is, here, I'll point to it right here, the bone that's right in this area, right here. Yep, here's my maxilla. Here's my maxilla. And I'll help you out with this one because it's a little hard to see. The one over on the side is called the zygomatic bone. If you look really closely, you can kind of see a dividing line between the maxilla and the zygomatic bone. Just know that the zygomatic bone is over on the side of the orbit. The maxilla is down on the bottom side of the orbit. So frontal bone in your forehead, nasal bone in your nose, maxilla making up your upper jaw, and the zygomatic bone that goes toward the side of your face. That helps us to make up your cheekbone. All right, now let's talk about the eyeball. The white part of your eyeball, do we know the name of that yet? What's the name of the white part of the eyeball? Or what's supposed to be white, right? Yeah, the part of your eye that's supposed to be white is called the sclera. I say supposed to be because, you know, it's pretty tough these days to have whites of the eyes that are actually white. My 4-year-old likes to say that my eyes are cracked. So sometimes she looks at my eyes and says, "Mommy, your eyes are cracked." I think that means bloodshot. So when I'm not sleeping enough, my sclera doesn't look white. But normally this is a thick connective tissue layer. It has a lot of collagen proteins in it that make it look white. So here's the white part of the eye. The colored part of the eye, which on this eye is blue, but it could be brown or green or hazel. What do we call the colored part of the eye? Do we know the name of that colored part? Yeah, the colored part of your eye is called the iris of the eye. And the iris can get bigger or smaller depending on the amount of light that you're exposed to. When there's a lot of light, your iris is going to look bigger. We'd see more of the colored part to make sure that not too much light gets into your eyeball. When it's really dark, your iris gets smaller because it actually pulls back toward the backside of your eye. Because the size of the iris, whether it's bigger or smaller, that's going to change the size of this opening right here. What's the name of the opening right here? Yeah, Jesse kind of hinted at it for us. Yeah, the pupil is the opening of the eyeball. So the pupil is the space where we see more or less light getting inside. The size of the pupil gets bigger or smaller as the iris expands or contracts. So to address your question, Jesse, when you're looking at describing, so the descriptions, matching up their names, probably the part in your lab packet that it talks about getting bigger or smaller is the pupil. But just keep in mind that the pupil is just an opening in the middle of the iris. So as the iris contracts more, that makes the pupil area get bigger. When the iris relaxes, it squeezes back down, and that allows less light to come in. The pupil gets smaller. Let's see. I think all that leaves for us then is muscles. Here's my wink, wink, nudge, nudge for you. You have more than enough other stuff that you're specifically memorizing. I don't want you to specifically memorize eyeball muscles. I want you to remind yourself what the words inferior and superior mean. I want you to remind yourself what the words oblique and rectus mean, because if you know what those words mean, I promise you, you can identify these muscles without having to memorize them. Let's work together and see if we can figure this out. I'm looking for a muscle called the inferior oblique muscle. Let's start with inferior. What does inferior mean? What's that inferior word? Yeah, exactly. Inferior is below. So I'm looking for a muscle that is below or that's lower. Do we remember what this word oblique means? What does oblique mean? Yeah, so oblique means we're at an angle. When I look at the bottom side of the eyeball down here, I have one muscle that is going parallel to or straight back. We'll see this better from another angle. Straight back following the optic nerve back there. And I've got another muscle that's going at an angle across the eyeball. Is it muscle number 1 or muscle number 2? That would be the inferior oblique muscle. Number 1 or number 2? Yeah, it's going to be number 2. Absolutely. So let's type its name here. Inferior oblique muscle. The inferior oblique muscle. It's at an angle. Hey, so check this out. I'm going to go up top. See this little thing up here that is at an angle compared to the eyeball? There's my superior. I'm going to put emphasis on superior oblique muscle. Superior oblique muscle. If I know that oblique means we're at an angle, and I know the difference between superior and inferior, I'm in good shape. I got this. We also have a superior and an inferior rectus muscle. When we talk about the word rectus, rectus meant parallel, or it's going the same direction as something. So when we look at this model from the top, you'll see that the superior rectus muscle and the inferior rectus muscle down here, they travel the same direction as the optic nerve does. They're parallel to the optic nerve. So the inferior rectus down below, I'll type its name really fast, inferior rectus. All that leaves for me then is this muscle and this muscle. Is it 1 or 2? That would be the lateral rectus. 1 or 2. Who's lateral? Yeah, number 1 over here, because remember, the nasal bone is medial. So number 1 over here is the lateral rectus. Lateral rectus muscle. And over here in the middle is the medial rectus muscle. Again, don't spend too much time memorizing the specific locations of these muscles. If we know superior versus inferior, if we know medial versus lateral, I promise you can predict the location of those muscles. Just use those directional terms. If you notice in your lab packet, when we look at this picture, it says that we're now looking at a lateral view of the model. If we're looking at a lateral view of the model, we can see, let's do some typing here, the zygomatic bone. I guess I should have put bone. The zygomatic bone over here on the side. we are seeing kind of a cross-section view of the frontal bone up here. I think those are our only bones, right? Yeah. Now from this view, you can see a lot better. See how this is a rectus muscle because it's running parallel to the optic nerve. So the optic nerve starts back here in the eyeball and extends back. I can see it over here. This is the lateral rectus muscle because I'm looking at the lateral side. We don't make you label it, but this little guy over here is the medial rectus muscle. Here's that superior rectus muscle. We cut a lot of it off up there. This that I see over here is what's left of my superior oblique muscle. Remember how we had an attachment that went over to the side before, and I'll show you on another view. But here's the muscle part of that attachment that we saw earlier, the superior oblique muscle up here. And here's that inferior rectus down below. One of the bone markings that we studied when we were talking about the face was a thing called the optic canal. When we looked at a skull, the optic canal was a circle opening for the optic nerve to pass through. When you're looking at this model, imagine that the bony tissue goes all the way across. So pretend what I just drew. Let's see, I'm going to make it orange. Pretend what I just drew here is a canal, or there's a little space where this bone extends over here. The optic canal is the space where the optic nerve goes inside of. So there's bony tissue that completely surrounds the optic nerve to keep it safe. The space inside, or this indentation where the nerve is inside of, that's the optic canal, where that nerve would pass through. This is the superior view of this model. Again, to help us orient ourselves in it, let me label some bones for us. Here in the right side of the model, we're looking at the frontal bone. We don't ask you to label it, but I'm going to mention the zygomatic bone is over here. So here's the zygomatic bone, the frontal bone. Right here is a bony structure that we learned that's an important one for us to be able to identify. All of those little dots that you see in there are holes, or they're foramen. So all those little dots that you're seeing are what we call the cribriform foramina. That was a bone marking on the ethmoid bone. So here's the ethmoid bone, which is in the middle of the skull. So labeling these bone markings, the goal of it is to help us realize that this is medial and this is lateral over here. So here's my medial side, here's my lateral side. That's going to make this muscle that I see right here the lateral rectus, and this muscle that's kind of tucked inside here, the medial rectus. Notice you can see very clearly in this view, here's that optic nerve. So the optic nerve that leaves the eyeball and travels back into the skull. So there's the optic nerve, all of my rectus muscles that are going parallel to it. That's why they're called rectus. Here's that superior oblique. You can see it at an angle across the top of the eyeball. I think, let me check my pictures here. Yeah, I think that's the last view of this particular model. Do we have any specific questions about this model? Go for it, Jesse.
Can you point out the ethmoid bone again? I missed it.
Yeah, so the cribriform foramina are actually in the ethmoid bone.
Okay.
So it's kind of all of this right here would be that all of this is the ethmoid bone with those cribriform foramina, the little dots that are inside of it.
Okay, thank you.
Uh-huh. Any other questions right now? Okay, so here's my reminder for you. With each of these views of the eyeball model, the first thing you should do when you look at that model is figure out which side is lateral and which side is medial. So when you're looking at the top view, if you can find that ethmoid bone that's here in the middle, that's the medial view, or the medial side, excuse me. The lateral side is out here because the ethmoid bone is in the middle. When we go back to this side view here, if we know that this is my zygomatic bone, because my frontal bone's over here, the frontal bone on this model is medial, that means I'm looking at the lateral view over here. And when I look at it head on, if I know the nasal bone is medial and over here is lateral, that's going to help me figure out the names of my muscles without having to memorize them. So the big thing I want you to memorize on this model, my markings that help me know lateral versus medial. It's all about the bones on this model. I point that out because my other eyeball model does not have bones. So we no longer have a bony structure to reference on this model. There are two ways you can tell lateral from medial on this model. Really the biggest one, the most important one, is going to be this structure right here. What is this structure right here called? Do we know? What is this thing right here? Yeah, exactly. This thing right here is called the lacrimal gland. Do we know from any review that we've done, what's the function of the lacrimal gland? What does the lacrimal gland do? Yeah, so the lacrimal gland makes tears. It makes the lubrication for your eyeball so that your eyeball doesn't dry out, so that your eyeball doesn't get infected. There's actually a very interesting article in Current Events in Anatomy. There was someone who was going through a very emotional time in her life, and she had a microscope at home, and she decided to collect her tears when she was crying for different reasons to see what they look like under a microscope. So if you're interested in seeing what your tears look like, the tears that you cry when you're sad look different from the tears that you cry when you're happy. It looks different from the tears that you cry when you're chopping an onion. So if you're interested in seeing what tears look like under a microscope, feel free to check that out in Current Events. But the place where all tears come from, or they all start from, is up here in the lacrimal gland. The lacrimal gland is found on the lateral side of your eyeball. So if you're looking at this model on the homework assignment or on the exam, the lacrimal gland is on that lateral side of the eyeball. That's your main structure to help you tell lateral from medial. I will mention too, though, that this little thing up here, this is what's left of the superior oblique muscle. Label that up here, superior oblique muscle. The superior oblique muscle always points toward the middle of the eyeball. So the direction that the superior oblique muscle is going is medial. The lacrimal gland is on the lateral side of the eyeball. With that information in mind, now you'll be able to find all of the muscles, or where those muscles should be. Notice that some of those muscles on your list, there's a little asterisk. We can't see them super well, but we can draw them on using our directional terms. So let me get a little red pen here. I'm going to draw a muscle right here. What would be the name of this muscle that I just drew? What would I call this one right here that I just added because it was missing? Yeah, that one over there is definitely on the lateral side because it's right there by the lacrimal gland, and it's going straight back, or it's parallel to the optic nerve. So I'd call it the lateral rectus muscle. Absolutely. So then I can draw the same thing over here on this side, and that would be called my medial rectus muscle. Hey, if I drew a muscle right here, what would I call that muscle up here? Exactly. I draw my little, here we'll add our little superior rectus. Superior rectus up here on the top. So again, lateral versus medial. That's the big thing we got to figure out. Once we know lateral versus medial, we can predict all of those other locations. Oh, my picture's gone. Okay, so that picture's missing. We'll just have to use this picture then. Draw it. Yeah, I need to figure out, if you give me a moment, I'll figure out which one it actually is, and I can draw it for us. I'll do my best. When I'm looking at this picture right here, I can't see the lacrimal gland, right? So I can't reference to the lacrimal gland to figure out what I'm looking at, but I can see my superior oblique muscle right here. That tells me, when I'm looking at this picture right here, which side of the eyeball am I looking at, if I can see that superior oblique. Yeah, so here is the medial aspect. That tells me that this one right here would be the medial rectus muscle. We already said this was the superior oblique, right? That's how I knew that we were on the medial side, because this one's up here, it's angling to the middle. So the superior rectus up above, the inferior rectus below, and the medial rectus up here. Okay, so I think I know. Here's my attempt at sketching. And it may not look like your picture because I'm guessing at what we're missing here, but let's give it a shot. And we'll add some muscles here. This looks real terrible. And I'm sure it's not, because I don't have a packet in front of me. So I'm making things up as we go here, but whatever. Okay, so I've got my, let's make my little lacrimal gland a little bit more full here. I got my lacrimal gland up here. Oh, thank you. You're too kind. I can see the lacrimal gland on the view of the one that we're missing here. The lacrimal gland tells me that we're looking at the lateral view, right? So the lateral view then shows me the lateral rectus muscle. Here's that superior rectus muscle, and here's the inferior rectus muscle. I added some stuff that we can't necessarily see on our pictures to help me orient it. This is the clear layer that's on the very front of your eye. Does anyone know the name of the clear layer that's on the very front of your eye? Yeah, so this is, we'll draw a line here. That clear layer on the front of your eye is called the cornea. That's the part, if for some reason your eyeball were to get scratched, you would scratch the cornea. So it's the outer layer of the eye. I added some blue here to represent the iris, just to give you a heads up. So this is the iris right here in my eyeball. Your model doesn't show it. And this is the cornea on the outside. So let me see. So Jacqueline's mentioning the white thing. And again, I don't know why it ate my picture. But the white thing is what we see right here. This white thing is what's left over of the superior oblique muscle. On this particular model, if we had the lacrimal gland, it would be peeking over there on the side. But we can't see it because we're on the medial side of the model. So this over here, the lacrimal gland would have been over here. This is the superior oblique muscle. Yeah, so the yellow thing that I drew and the yellow thing on your pictures is that lacrimal gland. Here, let me see if I can find that picture that it ate. Oh, see, I drew mine backwards. Okay, here, let me show you. I'll share my screen with you. I can't draw on it, but I can at least show you the picture that you have in your packet. Here. Here's the picture that, for whatever reason, my Blackboard ate. So here's that lacrimal gland up here. Here is if the lacrimal gland's right here, which muscle do we see right here? Yeah, this is the lateral side, right? So this would be the lateral rectus over here, since the lacrimal gland's right here. Lateral rectus right here, superior rectus right here, inferior rectus down here. I know a couple of you mentioned the little blue horns, right? These are actually blood vessels. So blood vessels leaving the eyeball, that's what those little blue horns are, just for your information. So lateral rectus, superior rectus, inferior rectus, and that lacrimal gland up top. Jesse asked a question I'll put to the class. Where is the sclera? Yeah, so any of the part on this image that's white is the sclera. The sclera is just the white part of the eyeball. So anywhere that's white, you can label as the sclera. Any other questions about this model? Any of those views of this model? Jacqueline says she likes this lab packet. Hey, we'll take it, right? Find the ones that you like, and then unfortunately you don't get to spend as much time studying the ones that are easy, right? You have to spend your time studying the ones that are not easy, alas. Okay, well, two more things that I need to cover with you. I need to cover the internal eyeball anatomy, and then I need to cover the retina with you. This picture is looking at the inside of the eyeball. So up to this point, we have just been looking at the outside of the eyeball. We've kind of been looking at it from this direction at the eyeball. Now we chopped the eyeball in half, and we're looking at the inside of the eyeball. The eyeball is actually made out of 3 different layers of tissue. So help me again in the chat. This outer white layer of the eyeball, what did we call that outer white layer again? Yeah, the outer white layer of the eyeball is the sclera. That's the outermost layer. The sclera is where all of those muscles that we talked about, the rectus muscles and the oblique muscles, the sclera is where all of those things attach. So outermost layer is called the sclera. Underneath the sclera, see this pink layer that's right inside here? This pink layer inside here is the thing called the choroid. Yeah, Hannah's right. That's the thing called the choroid. The choroid layer of the eyeball is the blood supply of the eyeball. Kind of like if you think about from lecture, we talked about the choroid plexus in the brain, how that was those leaky blood vessels. The choroid layer of the eyeball is the blood vessels that are inside the eyeball. The very innermost layer, or the most, I guess we'd call it deep, layer of the eyeball is the retina. So this yellow layer inside the eyeball is called the retina. The retina. Nicole asked a question about the choroid here. If this choroid layer was leaky, I don't believe it is. It's not just blood vessels. like the choroid plexus is. There's also, I believe, some connective tissue around those blood vessels and such. I don't think that the choroid of the eye leaks. It at least does not make cerebrospinal fluid, put it that way. Choroid doesn't mean leaky per se. I was mainly making the correlation that choroid meaning blood supply. So choroid plexus and the choroid of the eyeball both are blood supply. All right, so outermost layer, sclera, middle layer, choroid, innermost layer, the retina. And the retina is where you actually see. Now, the place in your eyeball where you can see most clearly is an indentation in the back. See this little indentation area back here? This indentation area back here is something called the macula lutea. The macula lutea. This is the place where your eyeballs naturally focus the light. This indentation called the macula lutea. So we see the most clearly here where we focus that light. Next door neighbor to it, area right here, see where there's blood vessels that are going into the eyeball itself? This space right here is called the optic disc. The optic disc. This is the blind spot of the eye. Because the blood vessels are coming in right here, there's no neurons there to collect light. So the optic disc is the blind spot of the eye. It's next door neighbors to the macula lutea, where we see things most clearly, where I focus that light. Behind the optic disc, that blind spot, is the optic nerve. So we can see all of the neurons that are part of the retina. They extend their axons out through the optic nerve back here that's leaving the eyeball. Nicole asked a question about the macula lutea, if there's more than one. Nope, there's just one in each of your eyeballs. So it's the space where when light comes in through the opening, that is the pupil, it's the space where that light gets focused, the one spot, the macula lutea back here. So just one per eyeball. They would be kind of in mirror image locations of each other when we look at the left eye versus the right eye, but there's just one of them in each of your eyes. All right, so we talked about these layers. We talked about the structures back here. Let's go back toward the front of the eye. What we don't see on our model, and it's something that I had drawn before when I drew my little sketch for you, we'll label it again. What's the name of the clear layer in the front of your eyeball again? What was that clear layer up here? Yeah, the clear layer up here is called the cornea. So the cornea is up here in front of everything. So most of your eyeball is covered in the sclera, except for the very front part of your eyeball where it's covered in the cornea. The cornea is clear, the sclera is white. I can see that colored part of the eyeball right here. What was the name of the colored part of the eyeball again? Yeah, colored part of the eyeball called the iris. So the iris is this colored part that I see right here. You have a note underneath your model asking you about the lens. We don't see the lens in this picture, but I'm going to draw for you where I would find it. So here's a little kind of lens shaped thing inside here. We're going to learn more about this in lecture, but the shape and the size of your lens gets changed when you're looking at something up close or something far away. So it's kind of like a flexible little disc that I'd find right here between the iris and this ridge that you see right here. This ridge that we see right here is called the ciliary body. So this ridge right here, the ciliary body. That's a ridge or a group of muscles that pulls on the lens to change its shape when you're seeing up close versus when you're seeing far away. I think that's everything in this model, right? Looking at that list there. Perfect. Okay, so one more thing that we need to look at is we need to zoom in close on this layer here called the retina. So imagine now that we zoomed in close on the retina. That's where we see this model here. When we look at this model, most of it, so most of this part up here, all of this is the retina. So here's the retina itself. Notice though underneath it, how I've got this layer that has a bunch of blood vessels. What was the name of the layer that was the blood supply for the eyeball? The blood supply of the eyeball? Yeah, the blood supply of the eyeball was the choroid layer. So all of these blood vessels that we see down here, the big ones and the little ones, all of this makes up the choroid layer of the eyeball, that pink layer before. This was all the yellow layer. And here on the very outside, all this white stuff that we see here was the layer of the eyeball that was the whites of the eyes. What was the name of the white part of the eye? Yeah, the white part of the eye is the sclera of the eye, the sclera. So we zoomed in really close. We mostly care about the retina, but we can also see the choroid and we can see the sclera when we look at this model. Inside the retina, there's actually 3 layers of cells. Down at the very bottom part of the retina on my model down here, these cells right here are the rods and cones. So this bottom layer of cells, they're shown here in blue, these are the rods and cones. The rods and cones are the cells that actually first detect light. Rods and cones detect light and they send all of their signals to a group of cells that are called bipolar cells. So this middle row of cells are cells called bipolar cells. Hey, not a trick question. We talked about bipolar cells in lab. What does that word bipolar mean? What did bipolar mean? Yeah, exactly. Bipolar means I've got 2 extensions coming off of my cell body. Check out this cell. We've got 1, this would be its dendrites, its cell body, and then 2, where it has its axon terminals. We've got 1 coming into the cell body and 1 going out, 2. My bipolar cells are all of these cells that I see here. They collect information from the rods and the cones. So rods and cones see light, or they collect light waves. They send it to the bipolar cells that are here in the second layer. This final layer here on the outside, all of these yellow cells, these are called ganglion cells. And ganglion cells are the type of cells that send vision information out of the retina and back to the brain. So ganglion cells are the ones that leave using the optic nerve to get out of the eyeball to go back to the brain so you can see things. Fun fact about vision, light waves actually come from right here. They go straight past ganglion cells, straight past bipolar cells, until they get to those rods and cones that are in the bottom layer of the retina. So in some ways, we kind of say that our eyes work backwards. Light waves pass all these other cell types before they get perceived, and then we send that information back up and out, which is kind of an interesting about them. Are there any last minute questions about vision? Got the crickets going on. All right, well, we will call that good then for the day. Here's something I'll mention. Since we are coming up on Thanksgiving break and you don't have any lecture assignments due until after Thanksgiving break, what you might want to consider is looking at the spinal cord lecture packet and the special senses lecture packet. Look at the parts of those packets that seem like they might match with what's going on with the lab packets, and maybe focus your attention over the next week and a half on the parts where lecture and lab really seem to match each other, because we have that lab exam when we first get back from Thanksgiving break. So you can kind of double dip, if you will, cover some lecture material that's related to the lab material. That'll help you do better in both classes. So consider maybe not doing what I call a linear approach. So going just all the way through lesson number 12, learning all the lesson number 12 stuff before you do number 13. Maybe do a little bit of bouncing here at the end to help you get ready for that lab exam. So tomorrow during our class, to answer Jacqueline's question, yes, we will have a live session tomorrow. We're going to start covering the spinal cord during our live session tomorrow, but consider over Thanksgiving break not only working on spinal cord, but also maybe doing some work on special senses, because I think that might benefit you in your preparations for the lab exam. So I'm going to go ahead and call it good on the recording. I will stick around for a few minutes here for any last minute questions. Otherwise, hopefully I will see most of you tomorrow at 9:30 for our lecture session.